Yeah,
It's a
okay.
slide rule.
So,
See, we don't even know what a slide rule is anymore. What is a slide rule?
no, a slide rule is what you used to have to make calculations. So I never actually used one. My dad did though. Um, I think it's a little bit like an abacus, that's used in China with all the beads. Yeah, it's the first calculator before calculators. So this is kind of the central dogma of molecular biology, in that there's DNA, it gets replicated into new copies. It also gets transcribed into RNA, and the RNA gets translated into a protein. So that's the way that's the progression of reactions. They're all really just chemical reactions, of course, and they're triggered by various signals. So here's an unraveling DNA, a double helix, as you can see. So one thing I'd wanted to point out for this one is that you've got cytosine there, and it's attached to guanine, and it's got 3 hydrogen bonds, but when adenine is attached to thymine, there's 2 hydrogen bonds. So that's why there are pairings only between guanine and cytosine. It's the nature of the nitrogenous base. So there's a lot of writing on these slides, so you won't have to take too many extra notes. DNA is double-stranded. It has a beta helix structure. The 2 strands are known as complementary. They're complementary in that one strand is a parallel replicate of the other strand, and not a parallel, but an anti-parallel replicate of the other strand. Adenine always pairs with thymine. Guanine always pairs with cytosine. RNA, on the other hand, single-stranded, forms some different structures. In the case of RNA, remember that uracil pairs with adenine, but guanine still pairs with cytosine. And you'll notice something else here that we'll get into, 5' and 3'. So what that's referring to is, guess what, the carbon. So, and where the carbon is. So when we're looking at this end of this molecule, look, here's the 5' carbon here. And if we're looking at this end, here's the 3'. But the anti-parallel nature,
sugar, right? That's the sugar.
that is the sugar, yes. That is the ribose, yes. And if we're looking at the anti-parallel strand, a 5' to 3', here's the 5' over here. And the 3' are all on this side. And of course, that goes for every molecule. So the 5' on this side and the 3' on that side on this strand, and the opposite is true of the other strand. And as it turns out, you can only add a new nucleotide to the 3' end. And you'll see why that's significant shortly. So DNA denatures at high temperatures, especially when the salinity is quite low. High temperatures, a non-neutral pH, a non-aqueous solvents like alcohols, for example, and interestingly, if there's a low guanine cytosine content. So, and one thing that we're doing in our lab later is we're going to make millions of copies of your DNA. And the reason we can do that is because of high temperatures. although we do have to use an enzyme from a bacteria that lives in a high temperature environment. So whenever we have one copy of DNA and we're going to replicate it, we're going to build a new strand based on the two old strands, like so. But of course, these strands here are held together by hydrogen bonds. You have to break them, and high temperature easily breaks hydrogen bonds. And that's very handy for us. And indeed, for the vaccines that are being made that contain RNA, they can be made with what's called polymerase chain reaction, which is the technique that we'll use for your DNA. And you can make loads of copies that way. Yeah, I'm not sure how the RNA one works, but it's probably somewhat similar. So that's making loads of strands of DNA basically in a machine called a polymerase chain reaction machine that separates these two strands here by temperature. And then when the temperature goes down, the new strands anneal together. They come back together. And then, oh, we would like to have more copies, if you don't mind. So in this case, this one will branch off, this one will branch off. And then we'll get new strands. So as you can see, you conserve the old strands along the way. That's called semi-conservative replication. So DNA is very interesting. A genome is all of the DNA in an individual. We all have genomes. Your genome's all your DNA, my genome's all my DNA. When they sequenced the human genome, I think that was 2000, it took like 10 years and lots of money. And now you can do it in a matter of hours. But we happen to have 6 billion bases. 6 billion bases. And when we're talking bases, it's referring to the nitrogenous bases, which is part of the nucleotides. 6 billion bases, or another way you can put that is 3 billion base pairs. Yeah, so mammal genomes, as you can see here, they're not, there's not a huge spectrum of sizes of mammal genomes. They're all very similar. Plants, interestingly, have an enormous variety in the sizes of genomes. They have some very large ones and some that are on the lowish side. Fungi, such as yeast, they have smaller genomes. They're a much smaller organism, of course. Bacteria, even smaller. Viruses have quite a variety, but they are small. So genome size normally correlates with complexity. So generally correlates with genome complexity. But there are some anomalies. There are some anomalies. The herpes virus, for example, has 230,000 base pairs, quite a lot for a virus. And some of the larger viruses, or sorry, some of the larger genomes such as the human genome have non-coding DNA. I think, how much is it? So like 98% or something is non-coding, approximately. I think it's something like 98% is non-coding of the human genome. And that probably means that the DNA that doesn't code for anything is a vestigial DNA from our ancestors. It's just not turned on by any signaling mechanisms. So the non-coding DNA stuff like gene families, intervening sequences that disrupt genes, satellite sequences, for example, those are contiguous blocks of repeating and repeating and repeating kinds of DNA. And that happens sometimes because sometimes accidentally during replication, pieces of DNA are duplicated more than once, maybe several times. And then the resulting daughter DNA can be much longer, although those duplicated bits of DNA don't code for anything. These are all eukaryotes. The prokaryotes have smaller genomes. Sorry, they're not all eukaryotes. Bacteria is a prokaryote. And of course, the viruses are considered a non-life form because they don't actually, they're not actually cells. So prokaryotes, bacteria, they have smaller genomes. They're often circular. Prokaryote genomes. And they have a single origin of replication and a single termination point. So their DNA then is replicated from here, it's the origin, to here. It's the termination, terminal end. So replication of a new strand is also double-stranded, of course. And replication got two new strands as you've replicated it. It doesn't really take that long, though. It just takes an hour, a couple of hours, I think, at the most, because it's a smaller genome. Can you imagine if the human genome, which is 6 billion bases, 3 billion base pairs, if you tried to replicate that from the beginning to the end, it would take a very long time. So that's not how eukaryote genomes are replicated. So bacteria also have something called episomes, extra chromosomal elements, and they're called plasmids. So in this case, these are the plasmids here. That's a plasmid. And that is circular DNA. And it turns out to be quite handy. So the main function of the plasmids is usually to code for antibiotics, like antimicrobial resistance to microbes, such as viruses. So viruses are always trying to infect bacteria. So they make antibiotics to kill other DNA and RNA that comes into the cell. So that's what the circular DNA generally codes for. Encodes resistant enzymes. What's very interesting is that there's this whole method now, and I'm sure you've heard of it before. I think some people call it CRISPR. There's different names for it. But here you've got the plasmids isolated from that bacteria. There they are there. And you've extracted, let's see, what color should that be? Extracted some DNA from a donor, a eukaryote. Could be a human. In one case, that's how insulin is made. So this could be, for example, e.g., the gene for insulin. And what's very interesting is that there are restriction enzymes that cut DNA at very specific sequences. So this is where these pieces have been cut, leaving little ends at each end of the segment. So it doesn't cut them exactly halfway. If you have two strands, like oh man, there's one strand. The other strand. But it doesn't cut through like that. Nuh-uh. Rather, it cuts like hang on, like that. So you end up having what are known as sticky ends, or little ends that stick out on either end. So on either strand, I should say, on either strand. So both of these strands have a little piece sticking out. And it turns out that you can take somebody else's DNA, cut it in a similar sequence, or at the same sequence rather, and then that DNA will anneal with any DNA that has the same sticky ends. So in this case, we've treated DNA with the same restriction enzyme as we treated the plasmid. And so our DNA now has these little sticky ends. And if we put them all together in a mixture with the plasmids and these pieces here, so this is our plasmid DNA, these are our donor DNA, if you mix them all together, then with another enzyme called ligase, that's a kind of a gluing enzyme, the plasmids will indeed take up some of the donor DNA. Sometimes the plasmids will just take up their own. So it doesn't work all the time, but it does work enough so that the transformed plasmids with the new DNA have what are known as a recombinant plasmid. So that's the bacterial DNA and the donor DNA. And that's great. So now you have the gene for insulin in the plasmid. You can put the plasmids back into the bacteria, and the bacteria will happily make insulin for you. And that is indeed how insulin is made.
Can you do that with yeast too?
Sorry?
Can you do that with yeast too?
Yes, you can also do it with yeast. Yeah, yeast also has plasmids, and yeast is actually better for eukaryotes. So there has been a transition from bacteria to yeast in a lot of cases because yeast has a eukaryotic genome. Yeah, definitely. So what does DNA look like? And this is really important because I'd like to introduce you to an avenue of genetics, molecular genetics, called epigenetics. So here you have a short region of the double helix. There, 2 nanometers. It's really tiny. So 6 feet of DNA will fit into one of your cells. And you saw how small your cells are. And they're teeny tiny. So you wrap, like the video showed, around a histone. You've got these beads on a string here. Looks like this in a micrograph. That's your chromatin. And then when the cell is going to be replicated, and your DNA, or the cell is going to be divided and duplicated, your DNA is going to be replicated, then you need to make chromosomes. They have to be packaged into chromosomes before mitosis. And so these are packed nucleosomes. That's just the chromatin wrapped around the histones. These are chromosomes that have been extended out. But eventually, before the cell divides, the DNA condenses more and more. So these are chromosomes that are in the metaphase of mitosis. They're quite condensed. And this is an entire metaphase chromosome here. Duplicated, of course, because it's been duplicated before for replication. Yeah, so there's a nucleosome that we saw in the animation, the fundamental subunit of chromatin. And in this case, the DNA was separated from the chromatin and dissociated to show the different sections. So each histone has different sections to it. They call it H, A, B, 3, 4. You don't need to memorize that, but that's just something interesting about histones. So it's not all packaged the same way during mitosis. So chromatin has 2 forms. There's euchromatin, which is compacted, highly compacted during interphase. Or sorry, not during interphase. No, I didn't say that right, sorry. It's highly compacted, but it is easily expanded. Compacted, but easily expanded to facilitate expression. So you can't go making messenger RNA from a compacted strand of DNA, not at all. So when a cell copies a DNA molecule, each strand serves as a template for ordering nucleotides into a new complementary strand. So I showed this before, but red and black. Here it is in blue. Here's the dark blue parent molecule. So the The molecule that's about to be replicated is called the parent molecule. The first step of replication is separation of the two strands. And as you saw in the video, that's done by an enzyme called helicase. That's the unzipper. And then each parent strand is now a template that determines the order of nucleotides in a new, what's known as a complementary strand. So the light blue here, those are the new strands. Eventually, then they're connected. The nucleotides are all connected. They form a sugar phosphate backbone. Of the new strands. And the new strands are called daughter strands or daughter molecules, which will have one parental strand and one new strand. So this is how it was discovered that it's semi-conservative replication. So when the double helix replicates, each of the daughter molecules will have one old strand and one newly made strand. How do we know that? Well, take a look at this experiment. Okay, here's a culture of bacteria. That contain N15 as the nitrogen that's in the DNA. It's N15, an isotope of nitrogen. And the other one has N14, which is a different isotope. It's a bit lighter. It's a bit lighter. So when you put something light and heavy into a centrifuge, the heavier piece will be lower down in the tube. The lighter bits stay up in the tube. So this is what happened. In the first replication, this is the first replication, the DNA was centrifuged. And then in the second replication, the DNA was centrifuged after 40 minutes. So this was a DNA sample centrifuged after 20 minutes, and then the second replication after 40 minutes. So let's look at the results. These are all the possibilities that could have happened. So it could have been a conservative replication, in which case the two new strands would be together and the two old strands would be together. But that didn't happen. It could have been that we've got one old strand here and one new strand. Same with the other one. That's semi-conservative, which is indeed what we have. But you wouldn't know just by getting one. Here you've just got one band. So clearly the weights are the same. If you had two new strands, two old strands, well, the old ones would weigh more because they come from N15. The new ones would weigh less because they come from N14. So you've got this band up here and you've got this band down there. We didn't get that. We got this. But you could also get that if the DNA, as shown here, is all, interspersed. So just like evenly interspersed. New nucleotides and old nucleotides. It would still all weigh the same. So what about the second replication? If you were to do a second replication, then in this case, you've got your two bands separate again. We don't have that. We don't have full conservation of the strands. Here we've replicated again. And it turns out that we have exactly what we expected. we've got our semi-conservative strands. If we replicated these, we would expect to find still only one strand. Not one strand, one band in our tube. All the same weight because everything is evenly dispersed. All our new nucleotides are evenly dispersed with the old nucleotides. So this one is the winner. Yay. Semi-conservative replication. So who carries out replication? Well, there's an enormous team that carries it out. For E. coli to be replicated, an hour or so for 5 million base pairs, a single chromosome divides to form 2 daughter cells. But a human cell can replicate 6 billion base pairs divided into daughter cells also in only a few hours. That's a considerably larger genome, however, and it's quite accurate. Only 1 error per billion nucleotides, which is amazing. There's a whole chunk of enzymes that are involved. When you replicate that long DNA molecule, start replication at what's known as an origin of replication. So in the animation, it just showed one replication fork at one origin of replication. In bacteria, it's a single sequence of nucleotides. The enzymes separate the strands, they form a bubble, and the replication proceeds in both directions until the entire molecule is copied. However, in eukaryotes, there are hundreds of thousands of origin sites per chromosome because they are so long. So at the origin sites, DNA strands separate, they form a bubble, and there's replication forks at each end of the bubble. So here is the origin of replication. This is the bubble. And daughter strands are formed at each bubble. And you can see that on a micrograph. You can see all the little bubbles. So in other words, you're replicating that molecule at the same time in many different places so that eventually when everything is replicated, you get your 2 daughter molecules as these bubbles expand. Eventually they fuse. Some of the enzymes are known as DNA polymerases. They catalyze the elongation of new DNA at the replication fork. The nucleotides align with complementary bases, and they're added to the growing end of the new strand by DNA polymerase. What is the rate? It's quite fast, about 500 nucleotides per second in bacteria, but only 50 per second in human cells. The raw nucleotides that are floating around waiting to be made into new strands of DNA are called nucleoside triphosphates. But each one of them has a nitrogenous base, a deoxyribose, and a triphosphate tail. So as each nucleotide is added, the last 2 phosphate groups are hydrolyzed. And they're hydrolyzed to form the 2 phosphates together known as pyrophosphate. And the exergonic hydrolysis of this little guy into 2 inorganic phosphate molecules is what drives the polymerization of nucleotides to the new strand. That's where the energy comes from. So here's your nucleoside triphosphate. It hydrolyzes. Here's your pyrophosphate. And when it does that, the new nucleotide is added. A new bond occurs. And you're probably thinking, well, you can't replicate DNA in the same direction at both strands. You just can't do it because the strands are anti-parallel. They run in opposite directions. So for this strand here, you can only add on to the 3' end. So you can only add nucleotides to free 3' end of the new growing DNA strand. So it can only elongate from the 5' into the 3' direction. That's a problem. That's a problem at the replication fork because one parental strand will be oriented 3' to 5'. The other one will be 5' to 3'. How do you do that? Well, in the animation that you saw, that one strand is a lagging strand, it's called, and it's replicated in bits and pieces. And that's the lagging strand. It's copied away from the fork in short segments. They're called Okazaki fragments. So the one that's replicated continuously is known as the leading strand. So in this case, this new strand is being replicated continuously. It can do that because this is the 3' here. It can be added onto the 3' of the new strand, no problems. But you can't do that with the lagging strand because it's running in the opposite direction. The new DNA has to be made in this direction. So you have to make bits of it. Bits of it here, bits of it there, another bit here. So each fragment's about 100 to 200 nucleotides long, and then they're joined by a ligase. Remember, the ligase is the glue. So that will form bonds, covalent bonds between the between the growing nucleotide strand. But interestingly, polymerases can't initiate synthesis because they can only add nucleotides to the end of an existing chain. That's another problem. Oh, man, it's one problem after another. So instead, to start a new chain, it needs a primer. You always need a primer, which is a short segment of RNA. It doesn't have any trouble attaching to that end. It's only about 10 nucleotides in eukaryotes. And primase, which is another enzyme, an RNA polymerase, links ribonucleotides that are complementary to the DNA template into the primer, that short chain, that short segment. And they can start that RNA from a single template strand. After the primer is formed, then the DNA polymerases can add deoxyribonucleotides to the 3' end of the ribonucleotide chain. Later, another DNA polymerase, a different one, replaces the primer. So eventually the primer, because it's RNA, not DNA, has to be taken out and replaced. And that's possible with this different polymerase, DNA polymerase. So returning to that original problem at the replication fork, the leading strand requires the formation of one primer only, but the lagging strand has to have a new primer when every new Okazaki fragment is made as the fork progresses. So that can slow down the process. After the primer is formed, polymerase adds new nucleotides away from the fork until it runs into a previous Okazaki fragment. In that case, ligase then joins the fragments together, Okazaki fragments. And of course, there are more players in our process. In addition to primase, there's those DNA polymerases and DNA ligases. There's other proteins that have a prominent role. One is helicase, that's the untwister or the de-zipper, unzipper. Yeah. So one thing that could happen is that you've separated the strands, but they start to reform, but you don't want them to reform before you've made your new strand. So there are some binding proteins that keep the unpaired strands apart while replication occurs. Yeah. So with the leading strand, you've got your primase for priming, putting on that RNA segment. Elongation, that all happens with DNA polymerase. A replacement of the primer, another DNA polymerase. With a lagging strand, you've got primers for each Okazaki fragment. That's added on by primase. Elongation happens with DNA polymerase. Replacement of the primer does happen with polymerase, but then you also need ligase to attach those Okazaki fragments together. So to summarize, the leading strand is copied continuously from a single primer, and the lagging strand is copied away from the fork in short segments requiring a new primer each time. So it's conventional convenient to think of the polymerase molecules as moving along a stationary DNA template, but in reality, the proteins involved in DNA replication form this large complex. I thought that was really well represented in that animation, a very large complex. It's anchored to the nuclear matrix. And they reel in, which I thought was shown very nicely, the parental DNA and extrude the newly made daughter molecules. So are there mistakes? Sure, there are. Just like making a mistake when you're typing. I'm sure you do. I know I do. Sometimes a lot of mistakes, and I have to go return, or erase, backspace, and make my corrections. However, interestingly, with your DNA, there are proofreaders. Wouldn't that be nice if you had just had somebody like a friend who was just always constantly proofreading your stuff? You didn't have to do it yourself. They just did it, fixed everything for you. Well, of course, nowadays with a Word and that does happen quite easily. You've got your grammar correction and you've got your spelling correction. In the case of DNA, enzymes proofread the DNA during replication and repair damage. Yeah, there can be mistakes. The wrong nucleotide, for example, like one error per 10,000 base pairs. Well, if that were the case, none of our DNA would work. But another DNA polymerase proofreads each new nucleotide against the template as soon as it's added. If there's any incorrect pairing, the enzyme removes the wrong nucleotide and resumes synthesis. So it turns out to be just 1 per billion nucleotides. And that one error usually doesn't make any difference whatsoever, because there's a lot of repeating of the codons, especially the last base of the codon. And DNA, of course, constantly subject to harmful chemicals and physical agents, radioactive emissions, reactive chemicals like nitrites, radioactive emissions, X-rays, ultraviolet light. They can change nucleotides in ways that can affect encoded genetic information. Sometimes DNA bases undergo spontaneous chemical changes, even just under normal cellular conditions. Some mismatched nucleotides are missed by DNA polymerase. There's some mutations that occur after the synthesis is completed. They can often be repaired, but sometimes they can't. But here's a mismatch repair. These are special enzymes. They fix incorrectly paired nucleotides. Yeah, sometimes that's just a hereditary defect. We talked about one, sickle cell anemia. That's an incorrectly paired nucleotide, just one. There's another that's associated with a form of colon cancer. Yeah. So what happens is excision. A nuclease cuts it out, cuts out the damaged strand, and then the polymerase fills in the cutout bit. So xeroderma pigmentosum, those are individuals that are very sensitive to sunlight, for example, ultraviolet light, and that can produce these thymine dimers. Dimers means 2 thymines instead of 1 between adjacent thymine nucleotides. So it buckles the DNA and it interferes with replication. So sometimes, because those mutations can be uncorrected in skin cells, it can cause skin cancer. So you're probably wondering, well, what happens when the replicating machinery, the polymerases, get to the end of the DNA? On one end, you're left with a 5' just hanging there. You can't actually add anything new on there. So there's no way to complete the 5' ends of daughter DNA strands. If you didn't correct for that, your DNA would erode. Every new strand would have a few less nucleotides. So there's your primer. You've taken it out. Whoops. You'd get shorter and shorter molecules if that were the case. But fortunately, the ends of chromosomal DNA molecules are called telomeres, and they have very special sequences, typically TTAGGG, repeated like 100 to 1,000 times. So they protect genes from being eroded because they don't code for anything. They're just there. They're telomeres. But there is even a mechanism to restore shortened telomeres by an enzyme, a fantastic enzyme called telomerase. It uses a short molecule of RNA as a template, and it extends the 3' end of the telomere. So that's fortunate. It doesn't repair the 3' end overhang, but it does lengthen the telomere. So there is some repair probable, but it's not present in most cells of multicellular organisms. So the dividing somatic cells do become shorter. That they're present, however, the telomerase, that neat enzyme, is present in germline cells, the cells that will become gametes, will become eggs and sperm. And active telomerase can also be found in cancerous somatic cells. So in cancer cells, of course, they replicate uncontrollably. If they replicated uncontrollably and there was new cells made all the time, eventually they would just run out of DNA to replicate because they would keep being eroded. But unfortunately, in this case, the telomerase can be present and it cancels out the shortening. Yeah, all right. Thank you for listening.